Hello everybody. In this video lecture, we will cover cell structure and function. So this is a PowerPoint that I will use. Let's go ahead and begin. So a cell is the smallest unit of a living thing. Cells fall into one of two broad categories: prokaryotic cells and eukaryotic cells. Prokaryotic means before nucleus, and eukaryotic means through nucleus. So prokaryotic cells are single-celled organisms of the domains bacteria and archaea. And domains are the biggest. So domains. Okay, so when you want to classify life, you divide it into different groups. We call it taxonomy. And the biggest group of a living organism is a domain. We have three domains: bacteria and archaea and eukarya, right? So this is like a biggest group of living organisms. And bacteria archaea includes eubacteria, archaebacteria, and eukarya includes what we think about living organisms, right? Plants, animals, fungi, and very diverse group of protista. So this is kind of like older classification, or it says traditional kingdoms. Now classification of organisms goes through different stage when they divide it into supergroups instead of kingdoms. But we will just look at this classification just for the purpose of understand of prokaryotic and eukaryotic cells, right? But we will briefly discuss these supergroups that are part of modern classification, if it makes sense, right? So if you look at any cell, right, you can classify it either being prokaryotic cells or eukaryotic cells. And if it's a prokaryotic cells, that those living organisms, they belong to domain bacteria and archaea. If it's eukaryotic cells, those organisms belong to either plants, animals, fungi, or protists. Prokaryotic before nucleus means these cells, they do not have nuclei, right? And nuclei are organelles within a cell. They do have DNA, however, because all living organisms have DNA. If you're looking at eukaryotic organisms, they made from eukaryotic cells, and those cells, they have nuclei, right? And bacteria and archaea, they are single-celled or unicellular organisms. When eukarya, most of eukarya are multicellular organisms. However, we do have some unicellular protists and unicellular fungi. So even if you're looking at prokaryotic cells, eukaryotic cells, any cells, they still have several basic features. So all cells, prokaryotic and eukaryotic cells, would have plasma membrane, right? And this is the boundary between external and internal environment of a cell. Inside this plasma membrane, you would find a thick jelly-like fluid called cytosol. And within this cytosol, you would find some suspended components of a cell. So it would be some organelles, either membrane-bound organelles in eukaryotic cells or non-membranous organelles, but organelles are inside the cytosol. Now, also all cells will have one or more chromosomes that carries genes made of DNA. And all cells have ribosomes. Ribosomes are organelles that build proteins according to the instructions from the DNA. So on this diagram below, you actually see this. Those are human cells, epithelial cells. They're taken from the covering of a cheek, internal covering inside the oral cavity, right? So those are cheek cells. They are eukaryotic cells, and they have nucleus. What have this dyed? It's dyed in a darker blue color. Those are nuclei. And these little dots that you see all around are actually bacteria, and bacteria are prokaryotic cells. So you pretty much can compare the size of the prokaryotic and eukaryotic cells. Prokaryotic cells are the size of organelles inside eukaryotic cells. And what's covered here in blue is a cytoplasm. Cytoplasm includes cytosol and all the organelles within it, right? And you can also see the boundary that is plasma membrane or cell membrane. Now, this is the diagram of a prokaryotic cell. It's a simple single-celled or unicellular organism that lacks a nucleus or any other membrane-bound organelles. So prokaryotic cells like this bacterium, for example, it would have, of course, cell membrane, as any cell has. It would have some type of cytosol, some fluid inside, ribosomes, those dots, and this is a chromosome. So chromosomes that has DNA and carry on genes. Bacteria might have a cell wall, it might have some capsule, it might have some extensions like a flagellum for movement, pili, but you can see it's pretty simple organization. And the area where chromosome is located is called nucleoid. That's a nucleoid region. A eukaryotic cell is a cell that has a membrane-bound nucleus and other membrane-bound compartments or sacs called organelles, which have specialized functions. So over here, you're just giving the size, proximal size of different cells and organisms. Right? So you can see like that's where we have eukaryotic cells, animal and plant cells, and here bacteria and mitochondria. Mitochondria are organelles within eukaryotic cells, and you see they have comparable size with the bacterial cell. Viruses are smaller, proteins and lipids and atoms are a smaller one, right? And then we have one of the biggest cells of an eukaryotic organism, an egg or ovum, and then they just show you the organism. And something that you can see only with electron microscope, some that you can see with light microscope and with the naked eye. So here's two diagrams of one diagram of two eukaryotic cells. That's an animal cell. This is the plant cells, and it's like a standard cell, right? So not every cell look like this. This is just give you an idea that eukaryotic cells, they have plasma membrane, and then they have all these organelles suspended within cytosol. Then this region between the nuclear envelope and the plasma membrane is called cytoplasm. And cells have nucleus, endoplasmic reticulum, rough, smooth, mitochondria, Golgi apparatus. Where did I show my? So this is a mitochondria, Golgi apparatus. These lines represent cytoskeleton. And also, if you look at a plant cell, you see the plant cell has a cell wall that animal cell is missing. Plant cell has central vacuole and chloroplast. So these three organelles are not found in animal cells. Okay, so we're gonna start with plasma membrane. Plasma membrane made up of phospholipid bilayer, which embedded proteins that separate the internal contents of the cell from its surrounding environment. So plasma membrane separates internal and external environment of the cell. A plasma membrane is a double layer, right? So this is one layer, here's another layer. And the main molecule is phospholipid. And then we have this shown here in the yellow. This is cholesterol. And what's shown here in big blue color, those are proteins. And what attach over here to proteins or attached to the lipids are sugar, right, or carbohydrates. So when carbohydrates are attached to lipids, we call it glycolipid. When sugar is attached to protein, we call it glycoproteins. Also, we have different type of proteins. We have integral proteins. This is transmembrane protein. So integral proteins, they kind of embedded within this membrane. And we have peripheral protein, let's say this one, look like peripheral protein. These lines here, because this is inside a cell, a cytoplasm, so this is cytoskeleton. So those are proteins, protein fibers that give cells stability, it gives it shape, allow for movement, movement of the organelles within the cell, right? So that's a plasma membrane. So plasma membrane is not just some barrier, it's a very dynamic and multifunctional organelle, because it's also allow for substances to move in and out of a cell. It allow other cells to recognize this cell. It's allow for communication. They can be enzymes. So they have plasma membrane has variety of functions. Now, cytoplasm. Cytoplasm comprises the content of a cell between the plasma membrane and the nuclear envelope. Cytoplasm is made of organelles suspended in a gel-like cytosol, the cytoskeleton, and various chemicals. Right, so cytoplasm pretty much everything between the nucleus and cell membrane. So many metabolic reactions, including protein synthesis, take place in the cytoplasm. On this diagram, we can see a plant cells, Elodea cell, and what you see is this green organelles, those are chloroplasts. You can see also cell wall, you can see the cytoplasm, that's kind of everything inside, plasma membrane. I don't know, it's kind of hard to see a nucleus, but there is a nucleus inside each of these cells. The cytoskeleton is a network of protein fibers. There are three types of fibers within the cytoskeleton: microfilaments, also known as actin filaments, intermediate filaments, and microtubules. And cytoskeleton provides mechanical support to the cell and helps a cell maintain its shape. So if you look over here, so this is fluorescent microscope picture, and what's shown here in these bright colors are part of cytoskeleton, and this is the biggest one, microtubules. Right, this is our actin or microfilaments, and this is intermediate filaments. Cilia and flagella are motile appendages that aid in movement, and they have the same basic structure. So cilia and flagella, they have the same basic architecture, but cilia are generally shorter and more numerous than flagella. So this is sperm cell, and sperm cell has a flagellum that aids in movement. And this is unicellular organism, a protist. It's an animal-like creature, and you can see these numerous cilia that allow this proteus to move. Over here, however, you see a cell that cover respiratory tract of a multicellular organism, and it also has cilia, but those cells do not move. These cilia just move substances along a cell surface, like a mucus and trapped maybe debris, maybe bacteria. Right, so cilia is for movement of entire unicellular organism or for movement of substances along the cell surface. So flagella are long hair-like structures that extend from the plasma membrane and are used to move the entire cell, for example, sperm cell or euglena. Here again, you can see sperm cells, and this big cell over here is actually an egg or ovum. And cilia, and singular is cilium, are short hair-like structure that are used to move entire cell as a paramecium, that is a type of a protist, or move substances along the outer surface of the cell. For example, cilia of cells lining the fallopian tubes that move the ovum towards the uterus, right? So, so over here, this is the cilia of fallopian tubes. However, those are individual cells, and they also have cilia. And I think this is the respiratory tract. So it can be respiratory tract, or it can be some other organs of multicellular organism that, that designed to move something along the surface of the cell. Endomembrane system. Endomembrane system is a group of membranous organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins. So it's a whole system made of different organelles and different membranes inside, and it includes nuclear envelope, so shown on this picture in a kind of orange color, lysosomes, here's the lysosomes, vesicles, so here's some secretory vesicle, transport vesicles, endoplasmic reticulum, so all this structure over here, and Golgi apparatus is this structure. Although not technically within the cell, the plasma membrane is included in the endomembrane system, because that's another membrane here. Nucleus. Nucleus, plural nuclei, houses the cell's DNA in form of chromatin and directs the synthesis of ribosomes and proteins. So in this picture, you see this larger circle, that's a nucleus. Inside the nucleus, you would find DNA in the form of chromatin. The nucleus is separated from the cytoplasm by a double membrane called the nuclear envelope. So this part over here, in blue color, this is nuclear envelope. And there are nuclear pores, like opening within this envelope, that allows materials to move between the nucleus and cytoplasm. The nucleus contains nucleolus, where ribosomes are made. So within the nucleus, we would find like a dense region, and this is where all those genes located for ribosome, for ribosomal RNA synthesis, right, and assembly of ribosomes. Stored in the nucleus are long DNA molecules and associated proteins that form fibers called chromatin. Each long chromatin fiber constitutes one chromosome. Constitutes one chromosome. The number of chromosomes in a cell depends on the species. So if you look at this diagram here, so this is DNA molecule, double helix, right? It's nucleic acid. And then this DNA molecule wrapped itself around proteins that called histones, and then we call this structure chromatin, because we cannot call it DNA anymore, right? Because it has some extra parts, like these proteins. So we need to come up with a name. So the name of this structure, combination of DNA and proteins, there is also a little bit RNA involved. So DNA and proteins, now we call it chromatin. Now, chromatin is a very loose structure. It's not condensed. And this is how DNA exists through the life of a cell. And only when cells are ready to divide, this chromatin will condense. And condensing, it's forming this structure, very distinct structure, right? Very condensed chromatin. Now, we cannot call it chromatin anymore, right? Because it has a different architecture. Now, we call it chromosome. But if you look, what is chromosome made of? It's made of loose chromatin. And what is chromatin made of? DNA and proteins. Endoplasmic reticulum. Endoplasmic reticulum is a series of interconnected membranous tubules that collectively modify proteins and synthesize lipids. However, these two functions are performed in separate area of endoplasmic reticulum. Rough endoplasmic reticulum for protein synthesis and smooth endoplasmic reticulum for lipid synthesis and metabolism. So on this diagram, you can see that's a nucleus. And you already know that nucleus is surrounded by nuclear envelope. Nuclear envelope continues with this tubular structure, right? So it's all kind of like membrane inside a cell, right? From nuclear envelope to this tubules, and then these tubules. And some of them has ribosomes attached. And if we have this endoplasmic reticulum, ER, with ribosomes attached, we call it rough ER. Because under light microscope, it has this rough texture. It appears like it has a rough texture. And then some tubules do not have ribosomes attached, and this is smooth ER. So it's one organelle, ER, but it has two different functions: protein synthesis, lipid metabolism, right? So protein synthesis in the rough ER, lipid metabolism in the smooth ER. So the rough, in rough ER refers to ribosomes that start the outside of this portion of the ER membrane. These ribosomes produce membrane proteins and secretory proteins. Some products manufactured by rough ER are dispatched to other locations in the cell by transport vesicles, sacs made of membranes that bud off from the rough ER. So this kind of like bluish part over here, this is cytosol, right? That's a fluid. This part is rough ER. It's rough because ribosomes are attached at the outer membrane. So the job of ribosome is to make this protein. So here's a polypeptide, a protein, and it's kind of deposited inside the lumen of rough ER. Now, these ribosomes that attach to ER, they're going to make proteins that will be transported out of a cell, or maybe will be incorporated in the cell membrane. So I'll show you in a second. Let's go back very quickly. You see these proteins, these blue proteins over here within the cell membrane. So where those proteins are made? Those proteins are made by rough ER. Right, so I'm going back. All right, so those proteins are made in rough ER. Also in rough ER, cells manufacture proteins that will be secreted out of a cell. So some of these proteins now surrounded by a vesicle, right? And it's called transport vesicles. And it will be delivered to a different location, to the surface of a cell. either will be incorporated within a cell membrane or secreted out of a cell, right? So those vesicles are bud off from rough ER. Now, smooth ER continues with rough ER, but has a few or no ribosome on its cytoplasmic surface. The SER function include synthesis of carbohydrates, lipids, including phospholipids, and steroid hormones, detoxification of medication and poisons, alcohol metabolism, and storage of calcium ions, especially in your muscle cells, right? So, so that's continuous with rough ER. No or very, very few ribosomes, and because it lacks ribosomes, the function cannot be protein synthesis because ribosomes synthesize proteins. So it synthesizes pretty much everything else: carbohydrates, lipids, steroid hormones, right, and so on. Now, Golgi apparatus works in partnership with ER and receives, refines, stores, and distributes chemical products of the cell. So you see this purple transport vesicle. This vesicle, I will go back, this vesicle is coming from rough ER, right? So vesicle coming from rough ER. Vesicles can come from smooth ER as well. So first, those vesicles will be headed toward the Golgi apparatus. Now, when this vesicle comes, the membrane fuses, and proteins or lipids or carbs, now they move from the vesicle inside the lumen of the Golgi apparatus. So Golgi apparatus receives those products from ER. Now within, what's gonna happen, within it will be stored or refined or modified, and on the shipping side of Golgi apparatus, now new vesicles will be formed. They actually call them secretory vesicles now, and they will move toward the plasma membrane, right? So it's like post office, right? So you get some mail, and then you store them first, right? And then you're gonna distribute them into different directions to be delivered to different locations. Lysosome. Lysosome is a membrane-bound sac of digestive enzyme found in animal cells. Lysosomes are absent from most plant cells. Inside lysosome, there are enzymes, and those enzymes break down large molecules, such as proteins, polysaccharides, fats, nucleic acid, worn-out organelles. So what is the function of lysosome? A function of lysosome would be to digest large organic molecules, and digest means break them into smaller molecules. So let's say this cell is getting some food, right, from outside. So the food first will be delivered inside a cell in a food vacuole. So that's vesicle of that vacuole will be formed from plasma membrane. But now cell need to digest this food. So lysosome will fuse with the food vacuole, digestive enzyme will be released, and they will digest this food, right? Another function of lysosome can be, for example, if you have an organelle that is not functioning, right? So it's a worn out organelle. You don't want to keep organelles inside the cells if they don't work anymore. So lysosome gonna fuse again with the vesicle containing this damaged organelle, release enzymes, and digest this worn out organelles. In single-celled eukaryotes, lysosomes are important for digestion of the food they ingest and recycling of organelles. So we already discussed this. Lysosomes also use their hydrolytic enzymes to destroy disease-causing organisms that might enter the cell. For example, your white blood cells, called macrophages, that part of your body's immune system, have this lysosome that can digest bacteria and viruses that this macrophage gonna engulf. So let's say you have a bacterium in your body. Let's say you cut yourself in your skin, you have bacteria entering your skin, then macrophages can migrate to the area of infection and they're gonna eat this bacterium. Macrophages will form this special extension called pseudopods. They're gonna surround the bacterium, forming a phagocyte, right? A phagocytic vesicle, right? So this will be our vesicle. Within will be bacterium. But what to do with this bacterium now? Well, this vesicle will fuse with the lysosome, digestive enzyme, digest this bacterium. And maybe whatever debris it has, so this vesicle can come to the surface, fuse, and through the process called exocytosis, remove this waste product. So vesicles and vacuoles are membrane-bound sacs that function in storage and transport. Vacuoles are somewhat larger than vesicles. Vesicle can fuse with other membranes within the cell system. So everything that is formed from ER, from Golgi apparatus, right, or from the cell membrane, that would be some vesicles and vacuoles. So over here you can see how protein is synthesized by ribosomes on the surface of rough ER, then its transport vesicle delivers this protein to Golgi, it moving through the lumen of the Golgi, and then another transport or secretory vesicles are formed, fuse with the plasma membrane and secrete this protein either outside of a cell or incorporated within plasma membrane. Ribosomes responsible for protein synthesis. Ribosome components are made in the nucleolus but assembled in the cytoplasm. Ribosomes may assemble proteins while the ribosomes are either suspended in the fluid of the cytoplasm or attached to the endoplasmic reticulum. Right? Ribosomes are not membrane-bound organelles. That's why ribosomes are found in both eukaryotic and prokaryotic cells. And ribosomes are made from protein subunits and RNA, a special RNA that is called rRNA, ribosomal RNA, and then some other parts involved like messenger RNA, transport RNA, and ribosomes gonna synthesize proteins. So ribosomes can be either attached to a rough ER, right, or they can be inside the fluid of the cytoplasm, inside cytosol. Chloroplasts and mitochondria are used for energy conversion. Cells require a continuous energy supply to perform the work of life. Two organelles that act as cellular power stations are chloroplast and mitochondria. Now, chloroplast only found in autotrophs, organisms that can use solar energy, right? They use this electromagnetic radiation or light energy or solar energy, and they use this energy to make actually sugar, right, carbohydrates, and convert light energy into chemical energy. So autotrophs are plants and maybe some algae, even some bacteria like cyanobacteria, they autotroph. So let's just, for example, take plants. So plants, they have chloroplast, and plants are autotroph, so they're capable of using light energy, and they use this energy to make sugar from carbon dioxide and water and convert solar energy into chemical energy. Now then plants have mitochondria as well. Plants are lucky, they have both. Animals only have one. Animals only have mitochondria, right? Plants have both. But now let's continue talking about plants. So when plants make sugar, they store this sugar. And when they need energy, they're gonna break down this sugar inside mitochondria and produce rich in energy molecule or energy rich molecule called ATP. And ATP will be directly used for work of life and for any processes needed to be done inside the cell. Well, not only ATP, but most of the processes inside a cell require ATP. So when cells break down sugar and they make ATP, they also make byproducts, carbon dioxide and water, and carbon dioxide and water used by chloroplasts to make sugar again. So that's like a cycle. Now, plants are self-sufficient. So they can use light energy, use chloroplast, make sugar, then use the sugar to make ATP, right, and continue this cycle. Animals, they need this sugar in their food. They need carbohydrates in their food, so they can break this carbohydrates, make ATP. So most of the living world runs on the energy provided by photosynthesis. Photosynthesis is the conversion of light energy from the sun to the chemical energy of sugar and other organic molecules. Chloroplasts are unique to the photosynthetic cells of plants and algae and the organelles that perform photosynthesis. So this process of the conversion of the light energy into chemical energy that happen inside chloroplast, this is called photosynthesis, right? So photosynthesis are unique for photosynthetic cells, and it involves the conversion of light energy into chemical energy of sugar and other organic molecules. So this green organelles inside a plant cell are chloroplasts. Chloroplasts are divided into three major compartments by internal membrane. So if you look over here, there is the outer membrane, right? And it also has, it's double membrane, so inner and outer membrane. And inside we have this green structure that is another membrane. This is called grana. All this grana made from several granum. So granum is singular. So that's a granum, granum, granum. So together it make grana. And the fluid inside is called stroma, right? And there is a space between two membrane, right? So the space is right here between inner and outer membrane of the chloroplast. Now, this granum is made of, that's shown here in green, this is called thylakoid membrane. So mitochondria are the organelles of cellular respiration found in almost all eukaryotic cells and produce ATP from the energy of food molecules. So this is electron microscope, and here's the mitochondria. Right? We'll look at its structure on the next slide. Also has two membrane, outer membrane and inner membrane. And this inner membrane makes this infoldings, right? Those folds goes inwards, and it's called cristae. So that's a cristae. And fluid is called matrix. And again, there is a space between outer and inner membrane. I'm sorry, maybe not a space. It wouldn't be right. It's a fluid within this, within the mitochondria. So this fluid is called matrix, and space is between the membranes. Also have fluids, of course, because everything surrounding by fluids. So mitochondria and chloroplast contain their own DNA, which encodes some of their proteins. This DNA is evidence that mitochondria and chloroplasts evolved from free-living prokaryotes in the distant part. So here's our mitochondria with the cristae and matrix, that's fluid, and this is chloroplast with the grana, and stroma is a fluid. Both have double membrane, space between outer and inner membrane, and both have their own DNA. Animal cells versus plant cells. Cell wall, a rigid covering that protects plant cells, provides structural support, and gives shape to the cell, only found in plant cells. Central vacuole, also found only in plant cells, plays a key role in regulating the plant cell concentration of water in changing environmental conditions. The liquid inside the central vacuoles provide tuber pressure, which is outward pressure caused by the fluid inside the cell. So cell wall and central vacuole, and of course chloroplast, are organelles found only within animal cells. Now, we already know function of the chloroplast, right? Function of cell wall is described over here. It's made of cellulose, it's very rigid, it gives plant cells support and shape. And central vacuole, shown here, is this central vacuole that is full of water, and that's why the plants look nice and healthy, because of this tuber pressure, outward pressure. So it pushes on a cell wall, and cell look like they full of water, and leaves nice and fresh. Now, when you stop watering your plant, right, it starts to wilt. And why it's wilting? Because you reduce the amount of water inside the central vacuole, and there is kind of like separation between the cell membrane or plasma membrane and cell wall. So cell wall and cell membrane are two completely different structures. Now, if we do not have pressure, we don't have this tuber pressure, we don't push this wall outwards, the plants look wilted. So let me see, is it the last slide? Yes, so this is our last slide for this video lecture. I want to remind you that we cover cell structure and function of different organelles. Thank you for watching, and I hope it was helpful.